Hi, everyone, and welcome to this month's Simon Says Experts Theory series. I'm Pat Loehrer. I'm director of the Center of Global Oncology and Health Equity at the Indiana University Simon Cancer Center. Clinical trials is probably more integrated in oncology than any other specialty. It's been over 50 years ago in 1971 when President Nixon signed the National Cancer Act. And it really signified the change of how we address cancer through clinical trials and other research. Over 40 years ago, Larry Einhorn and myself and Steve Williams, as well as a number of different community oncologists, formed what we call the Hoosier Oncology Group, or affectionately known as the HOG. And this provided clinical trials through patients throughout the state of Indiana. This group is going strong. It's now called the Hoosier Cancer Research Network, with over 200 sites around the country. Today, Kathy Miller joins me. Dr. Miller is a medical oncologist who specializes in breast cancer, and she also serves as the IU Simon Cancer Center's Associate Director of Clinical Research. She's truly one of the best breast cancer doctors in the country. She serves as a co-chair of the National Cancer Institute's Breast Cancer Screening Committee. She served as program chair for the American Society of Clinical Oncology several years ago and directs our cancer program here at the IU Simon Cancer Center. No one knows clinical trials better than Kathy Miller. Thank you, Kathy, for being here today, and look forward to talking about Clinical Trials 101.
Thanks, Pat. You know, this is a topic that is near and dear to both of our hearts.
Let's just start, if you don't mind starting out, clinical trials has a broad meaning. If you can explain in simple trials what a clinical trial means.
So clinical trials come in many different sizes and shapes, and they have many different purposes. And really, the best clinical trial starts with a question. And when you think about cancer care, you could ask questions of a lot of different types. You could ask whether this medicine to prevent nausea is more effective than a different medicine. You could ask whether treatments were differently effective. Was one better than the other? But you could also ask questions about whether a certain gene or protein in a tumor impacts people's prognosis. We might call those correlative studies. So the patients are getting the current best standard treatment, but we also get a sample of their tumor or a blood test to help us understand more about how different tumors behave. And those studies may pave the way for different treatments in the future. I think a common misperception that I'm sure you've heard is that clinical trials is only something that people think of when there's nothing left to do, when they've run out of treatment options. And we certainly do have clinical trials for people who've had lots of previous therapies. But there are also clinical trials for people who are just diagnosed with cancer because there are questions about the best treatments for people just diagnosed with cancer. And clinical trials are how we answer those questions.
We had a focus group many years ago with some patients, and one of the things they said is that they did not like the term trials. I don't know if you've had that experience.
I think that the terminology gets difficult. For some people, a trial sounds like we don't know what we're doing. We're just going to try it and see. But the term clinical experiment really doesn't go over it any better. So I tend to talk about it in terms of questions, that we've learned a lot about cancer, a lot about cancer treatment over the years. But as we continue to learn, it brings up new questions. And these clinical studies are how we answer those questions. And there may be times where the best treatment for you is not part of a clinical study. But there may also be times where being part of a clinical study that provides excellent care for you and your cancer, while at the same time helping us answer those questions, may be a fantastic opportunity.
You know, the other thing they bring up is I'm thinking about my patients, is that they don't want to be experimented on. They don't want to be a guinea pig. And then they ask, well, am I going to get a placebo? Can you talk a little bit about that?
So let's take both of those separately. So placebo is an inactive pill, a dummy pill, something that may look like exactly like the active drug, but doesn't have the active drug in it. And we would never in a clinical trial in cancer care just give people a placebo. But there might be times where a placebo is included in the trial. So suppose the question it is, we know that this current best standard of care works really well, but it's not perfect. And there's a new drug that we think might make that standard of care even better when added to the current treatment. So there might be a trial that compares the current treatment with the new drug or the current treatment with a placebo. So everybody's getting the current best standard of treatment, but some people might also be getting a new drug. And the reason you might want to use a placebo so that the patients and their doctors don't know who's getting what drug is we all have opinions. And our opinions about how well a drug might work, about what side effects it might have, could really impact how we think patients are doing and how they report side effects. So having the placebo gives us a better comparison, and we can be more sure about the answers to questions. That's a little bit different than people who say, I don't want you to be experimenting on me. And I respect that. Being part of a clinical study is not comfortable for all patients and in all circumstances. But we also have to recognize that if we can't do any more clinical studies, then the best treatments we have today are the best treatments we will ever have. I don't think we're okay with that. We still have a lot of work to do to cure more people, to do that with fewer side effects, to improve survivorship, to understand the disease. And those clinical studies are how we make progress.
Just to build on that, a lot of times I talk to patients about being experimented on. I tell them basically everything that we do is an experiment. If you have a pneumonia and we give you antibiotics, we're trying this antibiotic on you to see if it works. And most of the time it will, but many times it may not. I think much of what we do in life is an experiment. Even in testis cancer, in which you and I know is the really important here at Indiana University because of what Larry Einhorn did, has a 95% cure rate, and we know how to treat them. But still, there are patients who die from testis cancer, and so it's a low risk experiment every time we treat them. But our hope is that based on the previous results, that we're going to get some good outcomes from that.
I think it's also helpful for us all to remember that what we tell patients is currently the best available standard of care option, not long ago, was the new therapy being tested in a clinical study. That's how we learned that it was better, and it became the new standard. And I wouldn't be able to offer that to you if it were not for the women and men who were willing to participate in the clinical study that helped us learn that. I think it's also important, and I try to always remind patients that as much as I'm passionate about research, and I think that the clinical research is so incredibly important, my most important job is taking the best care of the patient who's in front of me. And if that's not being part of the clinical trial, I'm going to say that clearly. We may agree that a clinical trial is a great opportunity, but as she's being treated on that trial, if it's not working for her, if she's having more side effects than seems safe and reasonable, we're going to dump it and move on. Because sometimes we have to admit, it sounded like a great option. Once we got into it, we realized, it might be a great option for someone, not for you. We need to acknowledge that and come up with a better plan.
Yeah, I want to go back to the placebo a little bit. Much of what we have done in medicine is oftentimes based on dogma. This is what was done before, and therefore we'll continue to do this. I'm going back. This is even before your time, but back when I was a resident and I was working in the ICU, when patients who had ventricular arrhythmia after a heart attack, you'd look at their EKGs and the monitors as being VTAC, and of course you put them on lidocaine, which is a suppressant for the arrhythmias. Of course you would. Someone had the courage, if you will, or maybe the stupidity to do a trial called the CAST trial, which is the cardiac arrhythmia suppression trial. And what it actually showed is that those who were given antiarrhythmics did worse than those that were just observed. And I think this, to me, was a telling point about how we tend to think about things as being the logical, most practical thing to do. And then we do studies that show something different. I'm also, while I talk about this, I'm thinking about Bernie Fisher and his early work. And tell me a little bit about how we treated breast cancer 50 years ago and how it's being handled now.
Oh, it's the sort of thing that gives me nightmares. When you think about the early treatments, our surgeons did much more radical surgery. Everyone had a mastectomy. They removed the muscles under the breast. They removed as much skin as they could, as many lymph nodes as they could find. It was a horrifically morbid procedure. And fear of that surgery led to a lot of women not accepting screening, not coming in for evaluation when they found something different, because the treatment seemed honestly worse than the disease.
I'm going to interrupt you for a second. Just and the rationale behind that, it was sound in their mind. Halstead said cancer spread locally, and if you can get a wide margin and get around it all, you could theoretically do so. It was an honorable thought that you could get all the way around it, but it wasn't quite right. So I just want to interject. So go back to your story. Many
of the things we thought we knew made sense with our knowledge at the time when they were the best standards. And it sometimes takes real intellectual champions to propose a new idea, and then not just to accept their idea as, well, I used to think this and now I believe this. And clearly I'm right. They put their idea to the test. So Bernie Fisher was another very prominent, very forceful breast surgeon who thought Halstead's idea of this local kind of one step in front of the other spread was wrong. And that the tumors may spread directly through the bloodstream and not follow this logical stepwise order. And if that was the case, then removing so much extra normal tissue around the tumor may not improve survival at all. So he did one of the first randomized trials in breast surgery that compared doing a lumpectomy, so just removing the tumor itself and a little buffer zone of normal tissue around it, and then treating the rest of the breast with radiation, or doing the traditional Halstead surgery. And what they found was there was no difference in survival at all. And if anything, with long-term follow-up, we've learned that those patients who had a lumpectomy and radiation actually did a little bit better than those who had surgery. So it seems crazy now, the things that were believed then. But it also shows the power of research and the power of patients being willing to partner with us to answer those questions. And imagine the conversation with those women, suggesting to Mrs. Smith with a newly diagnosed curable breast cancer that not only may we be able to do less surgery, but we don't know. It's possible Bernie could have been wrong, and that doing less surgery would have meant less survival, greater chance of dying of your breast cancer. But imagine agreeing to be part of that study and letting a computer decide if you kept your breast or you lost your breast. It is amazing to me, the things that patients understand and are willing to help us address, the really important questions that they're willing to help us answer. And I think it points out that sometimes the barrier in clinical trials is us, in not explaining the reason for them, the importance of the question as well, or assuming patients won't do that, and not even asking the question, not asking patients if they will partner with us.
Yeah, I remember Bob Young years ago, who was head of the Cancer Center Director at Fox Chase and former ASCO president, said that with every clinical trial that he participated in, he knew the answer to the trial before it started, and he was right half the time. And I think that's the story, that we tend to think we know what it is until we get the results. We aren't quite sure. Let's talk a little bit about this randomization part. Why is it important to have randomized trial? Why is it important for a computer to pick one? Why can't we just pick which arm we want to be on for a trial?
Well, so actually, Bob gave you a potential answer to that, Pat. We all have opinions about what treatment might be better, what treatment might have fewer side effects, and our opinions might be wrong. Now, you and I are probably not wrong. People like Bob are wrong half the time. But the challenge with not having a randomized trial, with allowing patients and their doctors to just pick which treatment they want, is there can be some real biases in there. So you might give the new treatment to people who are younger, healthier, take fewer other medicines. They have less aggressive disease, less extensive disease. Those are people who are probably going to do better than older, sicker people who take more medicines, who have more extensive disease. And if you do that, you might then conclude that the new treatment is better, when it may not be better at all. In fact, it may be worse. You just picked people who were going to do better. So the randomization, having a computer pick which group patients are in, allows us to make sure that those groups at the end of the study are as similar as they can be on all of the things that we know might impact how well they do, and also things that we don't know about or can't measure. So if there's a difference in their outcome, good or bad, we can be much more certain that it's really because they got a different treatment. This has often been the case with new treatments, especially those that are more intensive, have a lot more side effects. They tend to be done first, the early studies in smaller numbers of patients, and they all get the new treatment, and we get really excited when they all do really well. But we have to remind ourselves all of the selection that goes into who gets access to those new treatments, and maybe not have quite the arrogance that we know the results of the trial before actually doing it.
You touched on it, but let's expand a little bit. There are different kinds of clinical trials, phase 1, phase 2, phase 3, and 4. Explain to the audience a little bit about the differences between these trials.
Sure. So those terms are typically used to describe the different steps in development of a new treatment. And the steps really are because we have different questions. So the very first question for a new treatment, a new drug, are some really simple things like, what dose do you give people in this drug? What are the side effects of this drug? How often do you give this drug? How can we give this drug to people safely? So phase 1 trials sometimes enroll people with lots of different types of cancer, because the side effects you have of a drug may not be different if you're getting the drug for lung cancer or colon cancer or breast cancer. And they often proceed a bit slowly and carefully because we want to make sure that we're keeping the patients who partner with us on these clinical trials safe. So they often will start at a lower dose. They'll enroll a small group of patients, follow them for several weeks and make certain that they're doing well, and then gradually increase the dose or look at different schedules. Once we've figured out a dose and schedule that is safe and tolerable, well, then the next question is, does it seem to work? Does this drug have some activity against cancer? Now, that question might be different. The drug might work really well for colon cancer and not at all for breast cancer. Or it might work really well in patients who've not had previous therapies, but not work so well in patients who've had previous treatments. So those are called phase 2 trials. They're a little bit larger. They can range anywhere from tens to 100 or so patients. And there are several of them asking, how well does this drug work in certain diseases and certain situations? The next step is what we call phase 3 trials. And that's really the key. Is this new treatment better than the current best available standard therapy? And depending on the treatment, that might be comparing this new treatment to the current standard. It might be looking at current standard by itself or current standard with the new treatment. So we have that direct comparison to see, is this really better? Now, you mentioned phase 4 trials, which we don't think about as much. But phase 4 trials are often done after a drug is approved, it's available on the market. But there are still questions that remain. And those may be, how well does this drug work in people who have some impairment of their kidney function or impairment of their liver function, or who are much older than the patients who were in the phase 3 trials. So those are often studies done more in a registry situation where everyone is getting the new treatment, but we're still collecting data on how well it worked for them and how the side effects were to make certain that there aren't specific populations for whom this treatment has unique problems.
Yeah, I consider the phase 4 more like Coca-Cola versus Pepsi-Cola. They're both out there, and some people prefer one or the other. And but when we think about, and when we're at the Cancer Center here, we have basic research, we have clinical research, we have population research, we have this term translational research. How does clinical research get informed? How do people, do they get around in a room and they say, okay, listen, let's just do a trial here in colon cancer? What's the process of the innovation and creation of a clinical trial?
And so sometimes it's a lot of serendipity. It makes it really important that we spend time together, that we talk to scientists who have different interests. Some of our best clinical trial ideas may come from our basic scientists who identify a new gene or a new protein that seems really important in how this cancer behaves. And they then work with chemists to develop a drug that might inhibit that gene or protein. And it's studied initially in cells growing in a plastic dish and then in animal models. But at some point, it has to move into the clinic and be studied in patients. So bringing people together may help those drugs make that leap. Sometimes it goes in the other way. We may have observations in the clinic about how well a drug is working, or it seems like it's not working in this group. We're having these side effects. And our conversations with our basic scientists may lead them to go back to the laboratory and change some of the experiments they're doing to try to understand why might that be. And some of the ideas are just chance meetings. When we happen to have an observation and you run into the right person in the hallway as that has just occurred and you're just asking that question or struggling with the results of that experiment or that patient. I was thinking as you were talking about Bob Young being wrong half the time, that a really well done clinical study teaches us just as much if the trial is negative, if our original idea didn't work, as if it did work. Now, I have been the PI of studies that have worked and of some that didn't work. It's a whole lot more fun leading a study that works. But if you've designed the trial well, you can learn just as much by seeing what doesn't work and then adding that to what we know about the disease and thinking about what did that tell us? What's the next step? What's the next question that we need to tackle?
I just want to remind the listeners, in fact, there may only be one listener. So the listener, if they want to send us any questions, put it in the chat and we'll try to do that. One question that did come up was that there seems to be an increasing emphasis on diversity and inclusion within clinical trials. How do you see the future of this fascinating topic and whether at IU Simon Cancer Center specifically or in clinical research as a whole?
So I think, and I hope that you're going to be hearing a lot more about this. So we've talked a little bit about drugs having different activity or different side effects in different groups of people based on the disease they have or how well their kidneys or liver function. But we need to start thinking about whether our genetic ancestry, the genes that we inherited, might also impact how well drugs work for us, what side effects that those drugs have. And in the future, those things may impact how we're treated. Now, this is not such a crazy question. So we do this already in medicine. If you're struggling with depression, your primary care physician is likely to order a blood test that measures several different genes that are inherited and how well they metabolize drugs. And that will then point them to the antidepressants that are more or less likely to be effective for you, rather than saying, we're going to try this one. We'll see how it works. If it doesn't work, we'll switch to another one. So we do this in other diseases. If you are on a blood thinner because of a blood clot or heart surgery. They've probably done a blood test to understand how well a blood thinner drug will work for you. And we are starting to think about that in cancer in ways that are really exciting and really powerful. And a lot of that work is happening at Indiana University. So I'll give you one recent example, Pat. So in the breast program, we started a little over 10 years ago recruiting healthy women who've never had breast cancer to come in for a breast biopsy, specifically so we would have normal breast tissue for research. And we initially thought that would be helpful in understanding how breast cancers first got started. So if you haven't clearly identified normal, identifying the first step outside of normal that ultimately becomes a cancer becomes an impossible task. So that was our initial goal. One of our scientists, Dr. Hari Natchatri, asked a very simple but profound question. Is the normal breast in women of African ancestry different than the normal breast of white women of European ancestry? And his question was really driven by, we know that Black women of African ancestry are more likely to develop an aggressive form of breast cancer called triple negative breast cancer. That had been known for over 20 years. We never had an explanation for why. And honestly, till recently, we didn't spend a lot of time thinking about, well, why is that? So Hari's really simple question was, well, maybe the cancers are different because the normal organ is different. And until Hari looked, everyone assumed the breast is the breast. The only difference is the color of the skin that's covering it. And it turns out that the normal breast is very different in women of African versus European versus Latina ancestry in ways that we think may explain the differences in the breast cancers they've developed. And that has given us some leads in how breast cancer in Black women of African ancestry might need to be treated differently. So we are about to do the first clinical trial, first clinical study, looking at one of those treatments. And for the first time, it will separately enroll a group of white women and a group of women of African ancestry. So we can not only see how well this treatment works, we can see, does it work differently based on ancestry? That's not something we've thought about before. And that's not something that we've ever intentionally designed clinical trials to look at before. But I think that will become much more common as we continue to learn more about the diversity of the human population and how those things have an effect.
Yeah, I'm going to append the question a little bit because of one of the questions from the audience. What kind of strategies are we using to create more diversity in studies and enrolling patients? What are we doing here to get more underrepresented people on trials?
Well, so I just gave you one. We also tried to make sure that all of the educational materials and all of the written materials that we have reflect the diversity of our patients and of potential clinical trial participants, that they respect the unique cultural aspects and how those decisions are made. We also spend a lot of time in the community, working with our Office of Community Engagement, helping people understand what are clinical trials, when would they be appropriate, why would you want to be part of a clinical trial. I also give all of my clinical trial patients a homework assignment. I ask them if they have felt like they were treated well as part of this clinical trial, they were treated with respect, they were glad they partnered with us on this clinical trial. I ask them to go back and tell their friends and their family, the people at their barbershop, their hair salon, at their church, because I know if they felt like they were disrespected and not treated well, and they were just a guinea pig in an experiment, they would be telling everybody who would listen. So if they've been treated well, I want them to be equally vocal to help gradually change some of those myths and stories that are still out there.
Yeah, as you know, we should not be proud of the fact of some of the experiments we've done in the past. Oppenheimer is out now, and I had some patients who were military officers who walked out to ground zero after the bomb went off. just because they wanted to know
They had
what
to
it looked
collect
like.
the metal to recycle it.
Yeah, and obviously Nazi Germany, but here in the United States, even into the 60s, we had this Tuskegee study that was done in the South, in which men and women were infected with syphilis, but not treated, well after that we knew that penicillin was there. And so this is part of the legacy of this. And I think particularly when we're dealing with African Americans, there's a hesitancy about going on studies, and because of the Tuskegee experiments there. And again, it's our obligation to recognize their reticence and understand why this is different, and why its communications are much better. We had another question in here. This is probably, I think the way I'm interpreting it says, when do you know it's time for a clinical trial, and who determines the timing? And I think this probably applies to a patient when they're coming in. How do they know they should go on a clinical trial up front or later on? Do they wait until they have recalcitrant tumor and then go on a trial? Can you talk a little bit about that?
So there are clinical trials in most of the circumstances that we deal with. So I think anytime that you need a different treatment, whether it's because you're just diagnosed and you need your first treatment, or you've had cancer for 2 decades and you need your 18th treatment, anytime you need a new treatment, it is the perfect time to ask, is there a clinical trial available for me? Would there be a clinical trial that matches my situation? Hopefully, your oncologist has asked that question and has talked to you about the best available standard option and clinical trial options that might be available for you. If they haven't, I think our patients should ask. Sometimes we need reminders. Sometimes it's a busy clinic and you're moving quickly, and you may have missed a clinical trial opportunity for patients. So it never hurts to ask at any time you're first diagnosed or anytime you need a new treatment. Many oncologists participate in clinical trials, but not all of them. And there's been some fascinating studies done that looked at the outcome of patients who were not treated on clinical trials. They were treated with current available standard treatment, but it compared people who were treated by oncologists who participated in clinical trials compared to oncologists in the same community who did not participate in clinical trials. And the patients who were treated at centers by oncologists who participated in clinical trials did better, even if they personally were not part of the trial. Now, part of that we have to acknowledge may be bias about who's able to access care at different centers. But part of it may also be clinical trials are looking at new ideas and new therapies. It requires you to stay up to date. It requires you to be thinking about the latest advances. It gives you experience with the new standard therapy before it becomes standard therapy. So you learn how to use those drugs and manage their side effects safely a bit sooner. All of those things may also have impacted those results.
I'm going to go into some of the other questions, but I want to probe a little bit more on this topic. There are oncologists who participate in clinical trials and those that don't. Why don't all oncologists participate in clinical trials?
It's a good question. I can't imagine not participating in clinical trials. But the reality is, it takes a lot of time and it takes a lot of work. Even if you're not writing the trial, you're not leading the national trial. I know that it takes 2 to 3 times the amount of my time in clinic to enroll a patient on a clinical trial, to treat a patient, manage a patient who's treated on a clinical trial, than to treat a patient who is receiving standard therapy. It also takes a lot of extra people. So it not only takes 2 to 3 times the amount of my time, I have research nurses, research staff who help with all of the documentation, who help manage those patients. It takes hours of their time. And when those people are doing all that work, someone has to pay them, and someone has to manage them. So there's a lot of infrastructure that is required in a practice in order to participate in clinical trials. And some practices have not made that commitment, and it's not available for them.
Yeah, I think they're special heroes. I think those people who are trying to make a difference in the world, and this is how you do it. One of the questions that was asked in terms of payment is, this is an expensive enterprise doing clinical research, and you're in charge of the clinical trials enterprise here at the university. Do participants need to pay to be on clinical trials?
No. Now we spend a lot of time looking at clinical trial budgets, and things that are only done because you are part of the study, or things that are done more frequently because you're part of the study, should be paid for by the study budget. They should not be charged to you or to your insurance. And as part of the process of joining a trial, of learning about a trial and deciding if you want to partner with us on that trial, your oncologist or their research staff should sit with you and explain that this is what's paid for by the study. These are things that would be part of your care, even if you were not part of the study. So they're still going to be billed to your insurance and to you. Now, there are sometimes some hidden costs that we have to acknowledge. Being part of the trial might mean that you need to see us in clinic more often, and that's more gas money, more wear and tear on your car, and the parking fees, and it might be additional time off work. Those things are hard to measure, and it's not like somebody hands you a bill for them. So we try to be clear about those requirements and those logistics. And we often will build into the study budget some reimbursement for patients for those extra visits to try to minimize their out-of-pocket costs or extra out-of-pocket costs. But there's no question that sometimes the extra visits and the logistics and the time required for that can be a real barrier for patients.
A couple of questions. One was, and I'll paraphrase this. At what point do you decide that a trial doesn't work, that you have this great idea, this great theory, and you keep pumping away at this idea and looking at different drugs and trying different iterations? At what point do you abandon this notion?
That's a really hard thing to do. I'll give you my thoughts, Pat, and I'd love your thoughts on this. I think it's much easier for me to conclude that someone else's idea and their trial is not working, and we ought to abandon it and move on. It is so much harder for me to have that same unbiased view of my own work. I think it's a little too easy to fall in love with your own ideas and your own trials. Part of the design of the trials includes working with statisticians who say, if this is your question, you need to enroll this many patients. And if you don't see this level of activity, this didn't work. You didn't meet that target, and the trial is done, even if you had hoped to go further. Part of it is something I think about more when I'm designing the trials. One of my favorite people in drug development said his goal was to provide as little ambiguity as possible, and that if an idea didn't work, he wanted to figure out that it didn't work and make it as quick and clean a kill as possible, because continuing to pursue an idea that's not working is wasting time and resources and effort that we could have devoted to the next idea that may work better. So I think this is one where it takes a village and other people reviewing our work to raise those questions, and we all have to be a little suspect to make certain that we're not kind of falling into that trap.
And, I was thinking of the high-dose chemotherapy, bone marrow transplant for breast cancer was around for many years. A lot of people treated with that. It was unethical not to do that. And yet, finally, the trials were done that showed that it just didn't work. And so it's interesting. The question is, are there certain types of cancers that are more predisposed to design of clinical trials? And if so, why?
I actually don't think so. I mean, we certainly have done clinical trials in all different types of cancer. I think where it gets difficult is in less common cancers. I alluded a little bit ago that statisticians are notorious for giving us hard numbers and saying to answer this question, your randomized trial may need 2,000 patients in treatment 1 and 2,000 patients in treatment 2. And if this is a disease that only 1,000 people are diagnosed in the US a year, that's going to be hard to do. So I think probably the only concern or the only real barrier would be in some of the rare tumors. That absolutely is not to say that it's not possible to do. And testis cancer is not particularly common. Thymoma is not particularly common. And yet IU has led clinical trials that defined and then redefined over and over again the standard treatment for those diseases. But that does require some unique resources and a lot of work with partnerships with other centers and other oncologists in different places.
Yeah, I'll put a little different spin on it. I do think that there are some cancers that are overrepresented in clinical trials, and that's largely because many of the clinical trials are designed by industry, and that they're going for market shares. And so lung cancer being the leading cause of cancer death in the country, breast cancer being the leading cause of cancer in women, are over predominant compared to, say, pancreatic cancer, which is there, but they so I think the industry pushes into looking at that and looking at drugs that may be more pertinent for those diseases. So that plays it. We have a few more minutes here. I wanted to
I
one
would
question,
agree with
which
you,
is
Pat, but it's not that the diseases are not predisposed to trials.
Right. Yeah,
there are
I
other
get
factors that impact what trials move forward.
Right. No, I agree with you. I think, but so this is a good kind of fun question. How have the clinical trials contributed to our field of oncology and the major treatments over the last 50 years? So I think what are some of the examples of what you think are the most impactful clinical trials over the last 50 years?
It is truly hard to think back about what cancer care was like for patients and their doctors 50 years ago. There was a recent analysis, actually, of the number of years of lives saved just by the results of large trials conducted by funded through some of the NCI large clinical trials networks. And it was hundreds of thousands of years of lives saved. So much so that I think it's hard to pick some of the seminal ones. I'll give you some in breast cancer. Understanding that blocking estrogen dramatically reduces the risk of recurrence after initial surgery in women with hormone-sensitive disease has saved thousands and thousands of lives with a therapy that for most women is very well tolerated and has a huge benefit. Identifying HER2-positive disease and developing therapies that specifically target that. More recently, immune therapy, especially in lung cancer. The idea that we would be talking about should patients with advanced lung cancer continue therapy? Do they still need to continue this therapy that they've been on for 5 years because they're fine, and what are we still treating? And even as early as when I was a fellow with you, the idea that we would be talking about 5-year survivors of non-small cell stage 4 lung cancer, that was not in anybody's wildest dreams when I was a fellow. And that is now a reality for some patients, certainly not for all of them. So the work is not done. But it is just such a different world because of the investment in basic research and the investment in then testing those ideas in the clinic. Do you have a favorite that's not thymoma, Pat?
Well, it's hard not to point to Larry's work in testis cancer back 50 years ago. Number one cause of death in young men, if you had advanced testis cancer, a 95% mortality rate. today it's a 95% cure rate for people with testis cancer. There's no tumor that has a high cure rate as testis cancer. It's amazing. I think I'm excited about, again, this link between basic science and the clinical realm, the work with Gleevec in terms of CML, and then translating that to this gastrointestinal stromal tumor, which is a relatively uncommon tumor. We used to have these patients with these cancers of the small bowel, and they would die quickly, and we didn't know what it was. And it turned out there was this rare subgroup. So we invented the disease. We didn't know what it was. And then we found out that there was a drug that hit it. Immunotherapy is another incredible story. As you talked about lung cancer, melanoma, it was hard to recruit a doctor that wanted to do work in melanoma because they all, if you didn't cure them with their surgery, they all died. And here you have Jimmy Carter, who it's been about 6 years ago, had brain mets from melanoma, and he was on immunotherapy, and he's, I think, the longest survivor on hospice. He's been on hospice now for about 7 months, I think. But the field of oncology is extraordinary. Just I'll close with one little note. In terms, this is Dave Johnson, who's a colleague of ours from UT Southwestern, presented a talk a few years ago and talked about the doubling of medical knowledge. And back in the 1900s, it was 150 years before medical knowledge doubled. It was mostly with pathologists describing diseases. In the 1970s, it was about 9 years. And today it's 2 months. There's a drug that's approved by the FDA each week for oncology indications. It's an extraordinary time. And I think all of this, underscores the importance of clinical research and the impact it has, and it's something that we're all involved with. So I want to thank you, Kathy, because we're running out of time here, but thank you for a wonderful conversation. And I want to thank all of the listeners for listening in and also proposing some questions that made this a much more lively discussion. But thank you for all of those who have been affected by cancer or are taking care of those with cancer. Our hearts are with you. And just to reiterate, we're on your team to make a difference. Thank you so much.
Thank you for having me, Pat. It's really been fun.